Hello, welcome to Bio 3. Today we will talk about Calvin cycle. Last time we talked about the light reaction of the photosynthesis. Remember, in the light reaction we split water. We use sunlight as an energy and split water so that it generates ATP and NADPH. Basically, Calvin cycle uses the ATP and NADPH generated from light reaction, and then we use these two energy molecules to so-called fix carbon. Carbon is from carbon dioxide gas, so we use these two energy molecules to reduce carbon dioxide, so that carbon dioxide will become carbohydrate, which is the sugar. That is the summary or overview of Calvin cycle. Okay, Calvin cycle. Here's some background information about Calvin cycle. It is also a Nobel Prize winning discovery by Mark Kelvin and Andrew Benson. They discovered that carbohydrate is made in the stroma. Remember stroma? Stroma, they are the fluid inside the chloroplast. It is using the Calvin cycle uses the ATP and NADPH from the light reaction, and also uses carbon dioxide from the atmosphere to reduce carbon dioxide to become carbohydrate. Remember reduction, what is the definition of reduction? Reduction is the gaining of, in this case, hydrogen atom. So when you put hydrogen atoms into the carbon dioxide, then carbon dioxide becomes carbohydrate, which is a form of sugar. Calvin cycle, okay? It's a series of reaction uses carbon dioxide from the atmosphere to produce carbohydrate. Okay, we repeatedly talk about this, but Calvin cycle is divided in three parts. This is important. Carbon dioxide fixation, carbon dioxide reduction, and then RUBP regeneration. So we'll go through these three parts one by one. Fixation of carbon dioxide, what does it mean? So carbon dioxide is attached to RUBP. RUBP is a 5-carbon molecule. When a 5-carbon molecule is added to one carbon dioxide gas, you have a total 6-carbon molecule. This 6-carbon molecule is then split in half so that it becomes two 3-carbon molecules. The 3-carbon molecule is basically called 3PG. Yes, 3PG, the word 3 at the beginning reminds you that 3PG is a 3-carbon molecule. So let me repeat. You have 1 carbon coming from the CO2, and then you have 5 carbon coming from RUBP, and then it becomes a 6-carbon molecule briefly, and then after this it becomes 3-carbon molecule, which is a 3PG, and then another 3-carbon molecule, which is also 3PG. So this is 3PG, And the reaction carbon fixation is accelerated by this enzyme RUBP carboxylase. This is the name of the enzyme required in the carbon fixation, carbon dioxide fixation, the first stage. So this looks very complicated, but don't worry, I will not ask you for the structure of each molecule. You don't need to know the structure. You just need to know the basic, the flow or the molecule, what are involved. So again, okay, you see that carbon dioxide here. Carbon dioxide is one carbon molecule. And then this is the RUBP. So RUBP. RUBP has how many carbon dioxide? If you remember from the last slide, it has 5 carbon It has 5 carbon atom. 1, 2, 3, 4, 5. So 5 carbon molecule RUBP is added by 1 carbon molecule, carbon dioxide. And then it becomes a 6 carbon molecule, which is a name that you don't need to memorize, but you just count the number of carbon. 1, 2, 3, 4, 5, and then 6. The 6th one is from carbon dioxide. And this reaction is catalyzed by RUBP carboxylase. Remember, RUBP carboxylase. This is the name of the enzyme that we covered from last slide, and then it has 2 more names. Okay, it just makes people confused. The original name that we told you from the last slide is called RUBP carboxylase, but now we give you another name. It can be called RUBP oxygenase. This is the second name. It can be called RUBP oxygenase. The last name is the easiest name. It's called Robisco. Okay, what does it mean, Robisco? Basically, it is the RU. You see the RU, and then BP, instead of calling it BP, it's just called bis. You know, bis is here. So it's RU bis. RU bis. What about CO? CO is the carboxylase. CO means carboxylase. So carboxylase is RU bis carboxylase. It's basically the same thing. RUBP carboxylase. Okay. Robiscos. So in the exam, I may ask you one of the 3 names. It can be it may be Robisco, it may be RUBP carboxylase, it may be RUBP oxygenase. This is the name. 3 names refer to the same enzyme. What does this enzyme do? It basically adds carbon dioxide into RUBP. The 5 carbon RUBP. Basically, that's the reaction. And then after this, the 6 carbon molecule. Oh, you don't need to know the name. Okay, too long, too complicated. Forget about this. And then hydrolysis. If you remember what hydrolysis means, hydrolysis meaning that it requires water to split the molecule. This 6 carbon molecule is split into 3 carbon molecule. And the 3 carbon molecule, so we can count 1 carbon, 2 carbon, and then 3 carbon. And it's called 3PG. 3 is for phosphoglycerate. So 1 carbon, 2 carbon, 3 carbon, 3 phosphoglycerate. This is what we have. We have 2 3PG. The PG is called phosphoglycerate. So that's the basic flow of the carbon dioxide fixation. We call it carbon dioxide fixation. carbon dioxide fixation, again, let's repeat. 5 carbon molecule, what's the name of the 5 carbon molecule? You just need to know BP. RUBP is the 5 carbon molecule added with carbon dioxide, so this is a 1 carbon molecule. 5 carbon plus 1 carbon becomes 6 carbon. And then later on it is split. The 6 carbon molecule is split into 2 3 carbon molecule. The 3 carbon molecule, what do we call it? 3 phosphoglycerate, 3PG. Okay, so that's all you need to memorize. Okay, RUBP add a carbon dioxide become 3PG. And then what is the enzyme that characterizes this reaction? Remember the enzyme name that I told you to memorize. The 3 names: Rubisco, bis, basically it's the PG, the BP is the BP, bis, and then co is the carboxylase, Rubisco, or you can call it RUBP carboxylase. RUBP, okay, carboxylase. LASE, or RUBP oxygenase, or so basically it's oxygen, and then you put ASE at the end, then it becomes oxygenase. So it's very, well, it's annoying. How come one enzyme has 3 names? But okay. In this case, you just memorize one of them. So in the exam, if I ask you the name, so you just give me one of the 3 names. It's fine. I suggest Rubisco is easier. The spelling is the shortest. This is what I memorized when I learned this photosynthesis. I just memorized Rubisco, that's all. Okay, this is the Calvin cycle reaction. We already covered this, carbon dioxide fixation. Now we will go ahead and talk about the carbon dioxide reduction. So let's take a look at the bigger pictures. So you see that RUBP carboxylase in here, yes, 50% of protein slow. I don't understand what it means. Seriously, some of the slides made by this Professor Mermovich, I don't understand what she talks about. Just remember that in carbon fixation, the one that we just talked about, it is the RUBP carboxylase, or you can call it Rubisco. It catalyzes this reaction. So it says that, okay, carbon dioxide is added to RuBP, so they become a 6 carbon molecule. 6 carbon molecule split into 3PG, which is a 3 carbon molecule. And then you can see that here you start with 3 carbon dioxide added to 3 added to 3 RuBP, so you get 3 6 carbon molecule. And then the 3 6 carbon molecule split into 6 3PG. Remember 3PG, they are 3 carbon molecule. So let me see. This is 1 carbon. This is 6 carbon. Then this is 3 carbon. So you have 3 6 carbon molecule, and then when you split in half, then you got 6 3 carbon molecule. So the number of carbon always conserved. You can keep track of the reaction by counting the number of carbon. And remember, okay, now we will go to carbon dioxide reduction. We have these, the ATP and NADPH, they are from light reaction. Remember, now we are using those, using the one that we invested. The goal is to make this something called G3P. G3P is the 3 carbon molecules, and it is a precursor to make glucose. The ultimate goal is to make glucose. Okay, once you go to G3P, you can assume that, okay, it is a precursor of sugar molecule glucose. Then you may assume that it is glucose. Okay, carbon dioxide reduction.